Hi everyone, my name is Uditi Desai, and I'm currently taking CBNS 106, which is an Introduction to Neuroscience class at the University of California, Riverside. So my current instructor is the lovely Yvette Gabriella, and we were assigned to do a video project for one of our final assignments. So I decided to do my topic on neurotransmitters. And in this video, I'll be discussing what neurotransmitters are, where they reside, as well as the different types of neurotransmitters. And I also wanted to touch base on some fun history facts that I learned in this class. And it's regarding the different theories of how the brain was connected, as well as the discovery of the first neurotransmitter, acetylcholine. So, have you guys ever wondered how the nervous system was connected? Well, this was a popular topic of discussion in the 1800s, and with this question arose two different theories. So the first was the reticular theory, which basically stated that all of the neurons are connected and fused together, and that's how messages got across in the brain. There weren't any gaps or synapses. And the second theory was the neuron doctrine, and the neuron doctrine basically stated that the neurons are separate and the cells communicate by contact of some kind. They weren't really sure what, and spoiler alert, it's kind of the title of this video, but yes, there were two main scientists, Golgi and Cajal, and Camillo Golgi decided to stain a neuron one day using silver chromate. And from his stain, he didn't think much of it. He saw that there were a lot of branching, and so he just assumed that there weren't any gaps between each branch. But later in the 1800s, Ramon y Cajal used Golgi's staining technique and decided to draw what he saw. And the results were shocking. So he saw that there were gaps between neurons, which solidified the theory of the neuron doctrine. And in the scientific community, there's a general consensus that the neuron doctrine is the more correct idea compared to the reticular theory. There is one point that reticular theory people really love to bring up, and it's the fact that electrical synapses create a continuum from neuron to neuron. But other than that, the general consensus is that the neuron doctrine is more correct. So Cajal was more correct compared to Golgi. So now we will discuss the journey of a neurotransmitter. And it all begins from neurotransmission. So basically, neurotransmission occurs when an action potential travels from the dendrites to the cell body, all the way down to the axon, and over here to the presynaptic neuron. And so the message causes the chemicals, which are the neurotransmitters, to be released from the presynaptic neuron into the synaptic cleft, and it will attach to receptors of the postsynaptic neuron. Now, this is really interesting. So the synaptic vesicles, they contain around 1,000 neurotransmitters. And for me personally, I knew that vesicles contain neurotransmitters. I just didn't realize how many there were in one tiny vesicle. So I thought when I learned that, I thought that was pretty interesting. So yeah, the neurotransmitter then travels across the synapse into it. Here's a synapse, and over here to the postsynaptic neuron, and it's like a lock and key. Like the neurotransmitter is like the key, and then the receptor is like the lock. It's a perfect fit. And from here, once it's attached, the message is passed on to the postsynaptic neuron. And the neurotransmitter can either excite or inhibit the postsynaptic neuron. And once it's done its job, it will just simply get released, and it can either diffuse out or get broken down or even go back into the axon of the first neuron. So, what are neurotransmitters finally? The answer is they are chemical messengers. You got mail messages. So like I said, neurotransmitters can either be inhibitory or excitatory. So inhibitory will inhibit, it will stop. It won't allow for any new action potential to process. And some examples of inhibitory neurotransmitters are GABA, glycine, and morphine. And there's also excitatory neurotransmitters, which excites the postsynaptic neuron and encourage and allow the action potential to be generated. And this allows more messages to be sent from neuron to neuron. And some excitatory neurotransmitters are glutamate and epinephrine. And neurotransmitters, they can also be modulators. And what this means is that it can basically enhance the inhibitory or excitatory responses to the receptor. And they don't directly activate any ion channels or anything. And some examples are proctolin and botulinum toxin, also known as Botox. And this makes sense because when we think about Botox, it prevents the muscles to contract, right? That's how it kind of prevents wrinkles, because how can you get a wrinkle when your muscle won't wrinkle? It won't contract. So yeah. So whether a neurotransmitter is excitatory or inhibitory, it all depends on the receptor it binds to in the postsynaptic neuron. So like I mentioned, neurotransmitters can be both excitatory or inhibitory. It all depends on the context. And another fun fact, neurotransmitters are endogenous, which basically means that they're produced inside the neuron itself. So most neurotransmitters, they're either amino acids, amines, or peptides. And this list, I believe my professor got from our textbook. And this list is just the tip of the iceberg for the amount of neurotransmitters there are. There are so many more than this. And in this class, we only highlighted a couple and discussed those couple neurotransmitters. So we know that there are 20 amino acids, and as we can see, not all are listed, but within those 20 amino acids, only 8 of them are neurotransmitters, and that's pretty interesting. But yeah, all 8 aren't listed on here, just 3, and within those 3, we just talked about these 2. So just to review, I wanted to provide the structures of the amino acids, amines, and peptides. With amino acids, we know they have the amino group, the R side chain, and carboxyl group. And amines contain the nitrogen atom with one or more hydrocarbon groups. And peptides are basically like chains of amino acids. And here are some examples of each one. So some example of amino acid neurotransmitters are GABA and glutamate. For amine, acetylcholine, dopamine, and epinephrine. And for a peptide, it is substance P. So one of the neurotransmitters that I wanted to talk more about was acetylcholine. And when we learned this, I was very much intrigued and did more research on this topic because I just thought it was really interesting. And that is the discovery of acetylcholine. So basically, there was a scientist named Otto Loewi and he did an experiment on a frog heart where he stimulated the heart with like electrical impulses through the vagus nerve, which, by the way, is the longest cranial nerve. And then he stimulated the frog heart, and then he used that old like bathing solution and transferred a new heart. And this did not receive any electrical impulses at all. And he noticed that the second heart, no electrical impulses, just the same like bathing solution, that heart rate was slowing down. And this is how he discovered acetylcholine. So at first he called it the vagus stuff, and I believe it's because like the vagus nerve, vagus stuff. And so now like the modern version of that is called acetylcholine, and I thought that was pretty interesting. So acetylcholine is found in both the central and peripheral nervous systems. Choline is taken up by the neuron when the enzyme choline acetyltransferase is present. Choline will combine with coenzyme A to produce acetylcholine. And the synthesis usually depends on whether or not these are available and how active the neurons are. So acetylcholine controls the heart rate, motor movement. It's distributed throughout the brain. It has diverse functions like cognition, motor functions. And one thing that was interesting was that Alzheimer's disease is caused because of the degeneration of acetylcholine in the basal forebrain. So although acetylcholine receptors respond to acetylcholine, they can also respond to nicotinic, and muscarinic receptors. So nicotinic receptors are ionotropic, and muscarinic are metabotropic. And ionotropic, the nicotinic ionotropic receptor usually generates and excites the neuron, and muscarinic, it honestly just depends on the receptor. So yeah, this is basically my presentation. I talked about how neurotransmitters go from the presynaptic to the synaptic cleft and then attached to the receptor of the postsynaptic neuron. And they are chemical messengers. There's 3 different types: amino acids, amines, and peptides, and they can be excitatory, inhibitory, or they can be modulators. And I talked about the history of acetylcholine, how it was all discovered through an experiment with frog hearts by Otto Loewy in, I believe, 1921. Yes, 1921. And yeah, this is my video presentation for CBNS 106. I hope you all enjoyed, and thank you for your time, and thank you for listening, and I hope all of you guys have an amazing day.